Hello and welcome you all to my channel Omni Bio Entrance, and today we will be discussing IAT previous year question paper conducted in the year 2024. I will be discussing only the biology section. If you are new to my channel, please do like, share and subscribe. If you want still previous year's question papers of IAT biology being discussed, you can refer to my channel Omni Bio Entrance. Go to the channel playlist and you will find it there. Coming to the first question, what will be the sequence of RNA synthesized using the following DNA template strand? So they have given the template strand here. The complement to the template strand will be your RNA, but the direction here is in the 5' to 3' direction in the answer. So be careful when choosing the options. So 5' I have written it here, 5' GTC DTAGG CTT CTC 3'. Your RNA sequence will be CAG AUC CG AA GAG, that is in the 3' to 5' direction, but the options they have given as in the 5' to 3' direction, so it should be from here, that is 5' to 3', this direction. So it is GAG AA GCC UAG AC. So the answer here is option A. Next question. The following pedigree diagram shows the inheritance of a rare genetic disorder. Filled shapes depict affected individuals. Which of the following is the most likely pattern of inheritance of the disorder? So if you see this, these are the individuals, these black ones are the individuals that are affected, whereas the others are unaffected. Here the female is affected, the female and male are affected. There is no skipping of generation. In all generations the disease is being shown, so it has to do something with the dominant condition. So it is not recessive. So autosomal recessive it cannot be, because autosomal recessive condition it will show skipping of generation. That is, if in one generation it will show the disease, then for the next generation it may not show the disease, then again the next generation it will show the disease. That is called as skipping of generations. Here both, so it cannot be autosomal recessive. Here both the individuals are affected, both male and female are equally affected. Then all affected males, they have an affected mother. So this male is affected, mother is affected. This male is affected, mother is affected. Okay, now here if you see, the male is affected and only the female child is getting affected by the disease, not the male child. So that shows that it has got something to do with X-linked condition. It is not autosomal dominant also. If it was dominant, then male child also would have got affected in this condition if the father was affected. But only the female child is getting affected. So it is not autosomal dominant. Now we will see whether it is X-linked dominant or X-linked recessive. Now, since it appears in all the generation, it has to be X-linked dominant. And we will confirm it that it is X-linked dominant again by doing the analysis. So here if you see, now if it was X-linked recessive, so X small r, it has to be there in both the X chromosome, only then it will be recessive. The female is affected, male is unaffected. So XY, these are the gametes, these are the offsprings. So offsprings, if you see, the female is the carrier, but here the female is affected and male is affected, male is affected. So it cannot be X-linked recessive condition. Now if you see X-linked dominant, it can be of two types, with both the X chromosomes carrying the defective gene or one X chromosome carrying the defective gene. Still then the offsprings will be affected. Now we will see whether it is present on both the X chromosomes or single X chromosome. Now if it is the case with both the X chromosomes, Then this will be the gamete for female, this will be for the male. The female will be affected, the male will be affected. That is, all the children are going to be affected if it is XR condition. So here only one female is affected, the other female is unaffected, one male is affected whereas other male is unaffected. Here also it is showing the same pattern. Okay, so it cannot be capital both the X chromosome carrying the dominant gene. We will see in case of single X chromosome carrying the dominant gene. Now if that is the case, this will be affected, this will be the gametes. So you can see that this one female is affected, one female is unaffected, one male is affected, one male is unaffected. That is the condition seen here. Some males are affected, some females are affected, some males are unaffected, some females are unaffected. Now if you take this condition where the male is affected, till now it was the female affected and giving rise to offsprings. Now the male is affected, the female is unaffected. Now male is affected means XRY into XX. So XRY will be the gamete, X will be the gamete here. You will see that the females are affected but the males are unaffected. That is the condition seen here. The female is affected but male is unaffected. Here also the male is affected, females are affected but males are unaffected. So this is a clear case of X-linked dominant condition. Then next one. Match the list of conditions in column 1 with the list of physiological process. Allergy. Allergy is exaggerated immune response to environmental substance. Everybody knows that. So P2. Uremia. Uremia is buildup of urea in the blood. It may be due to failure or impaired kidney function. So uremia means, okay, fourth one. Malfunctioning of kidneys which can lead to urea accumulation in the blood. Q4. Myasthenia gravis. It is an chronic autoimmune disorder that affects the nerve muscle connection leading to muscle weakness and fatigue. So R will be 3. It is an autoimmune disorder affecting the neuromuscular junction. Acromegaly. Next one. It will be due to the excess production of growth hormone by the pituitary after normal growth has stopped. So S will be 1. Excess secretion of growth hormone. So the answer here is option A, that is P2, Q4, R3, S1. Then next one. Which of the following proteins play direct role in muscle contraction? So the answer here is troponin. Troponin, it regulates the interaction between actin and myosin filaments in muscle contraction. Insulin, if you see, it regulates glucose metabolism and cellular energy storage. Then myoglobin, it stores oxygen in the muscle, and trypsin, it is an enzyme that breaks down proteins in the intestine. So the answer here is troponin. Then next one. Which of the following is not derived from epidermal cell layer in plants? So Casparian strips from rice roots. That is the answer. Option A is the answer. Why? Because they are derived from the endodermis. Casparian strips are derived from the endodermis, not from the epidermis. Then trichomes, they are hair-like structures found in the leaf derived from epidermal cells. Then subsidiary cells from rice leaf. Subsidiary cells encircle and regulate the stomatal aperture. They are the subsidiary cells. They are also derived from epidermal cells. Then bulliform cells. Bulliform cells, they roll up, help the leaf to roll up during drought or stress, thereby reducing water loss. And when water becomes available, the cells absorbs water, causing the leaf to unroll back. That is bulliform cells. So then they are all derived from epidermal cells, whereas A, that is Casparian strips from rice root, it is derived from endodermis. Then which of the following statement about meiosis in sexually reproducing plants, they have asked for plants, is incorrect? The end product of meiosis 2 are haploid gametes. That itself is the wrong answer. It is not haploid gametes, it can be haploid gametophytes. Because if you take the case of microsporogenesis and megasporogenesis, microsporogenesis meiotic reduction result in the formation of microspores or pollen grains, and each microspore creates the male gamete. Then in case of megaspore, the megaspore mother cell, it will undergo meiosis and give rise to 4 haploid nuclei. Then out of these 4 haploid nuclei, 3 degenerate and 1 functional megaspore undergoes meiotic division to give rise to embryo sac, which is then mature into a female gametophyte. Inside the embryo sac you will find the egg cell. So it is giving rise to megaspore and microspore, not to haploid gametes. These megaspore and microspore are the haploid gametophytes that give rise to haploid gametes. So the answer here is option A. Now B, if you see, the 4 products of meiosis are genetically different. Yes, there is recombination taking place. Crossing over and recombination taking place in the prophase 1 of meiosis. And there is independent assortment also. Then meiotic recombination take place both in males and females, of course. Then in most flowering plants, only one of the 4 products of meiosis survives in the female. Yes, that is also correct. Then next one. Which of the following graphs represent the correct relationship between light intensity and rate of photosynthesis? So if you see graph A, you can see that when there is increase in light intensity, suppose X is light intensity and this Y is rate of photosynthesis. If you see that as light intensity increases, rate of photosynthesis also increases, but when it reaches saturation, there is no further increase. So the answer here is option A. Then next one. Match the enzymes in column 1 with cellular components in column 2. So succinate dehydrogenase, where it is found? It is found in the inner mitochondrial membrane. Then pyruvate dehydrogenase, it is found in the mitochondrial matrix. Lactate dehydrogenase is found in the cytoplasm. ATP synthase is found in the thylakoid membrane. So the answer here is option A. Then next one. 2 species of flowering plants, Pn is equal to 20 chromosome and Q 30 chromosome, are reciprocally crossed with each other as male or female as shown below to produce F1 seeds. Okay. So which of the following seed tissues from both F1 seeds R and S will have the same chromosome number? Now what is reciprocal cross? Reciprocal cross is, first plant P will be the male and plant Q will be the female. In the next cross, plant Q is going to be the female and plant P is going to be the male. So whichever was the female parent in first cross will become the male parent in the second cross. That is called as reciprocal cross. Okay. So from there you will get F1 seeds. They have given the 2n value. And which of the following seed tissues from both the F1 seeds will have the same chromosome numbers? So is it embryo, endosperm, embryo and seed coat, embryo and endosperm? Okay, we will see. Now first, Cross 1. P is male, Q is female. 2n is equal to 20, 2n is equal to 30. This is embryo. N is equal to 10, n is equal to 15. The total will be 10 plus 15, that is 25 will be found in embryo after cross 1. Then in cross 2, P is going to be the female and Q is going to be the male. So 2n is equal to 20, n is equal to 10, 2n is equal to 30, n is equal to 15. So 10 plus 15 will be 25. So both numbers, that is 25, is the same. So it is going to be the embryo, that is your answer. It cannot be endosperm. Endosperm is 3n. An endosperm is formed as a result of the fusion between one sperm nucleus from the male parent and two polar nuclei from the female parent. So 2n is going to be your female parent and n is going to be your male from the male. So cross 1, P is male, Q is female. 2n is equal to 20, n is equal to 10. The male parent will be n. 2n will be your female parent. So 2n will be 30 in case of Q is the female parent. So 10 plus 30 will be 40. Now in cross 2, P becomes the female parent and Q becomes the male parent. So 2n will remain 20 there for female parent. And for Q it will be 2n is equal to 30, n is equal to 15. So 20 plus 15 will be 35. So here both the values, it is not the same. 40 and 35, they are not the same. So it is not going to be your endosperm. It cannot be your seed coat because seed coat is formed from the female parent. Okay, here the female parent will be different. 2n will be 20 in one case, 2n will be 30 in the another case. So the female parent is different. So seed coat number also will be different. Embryo and endosperm. Endosperm is not the answer. So the answer here is option A, that is embryo. Then next one. Which of the following is routinely performed to detect typhoid? It is Vidal test. Answer is option A. Then next one. Which of the following plasmid vectors can be used for cloning a gene with restriction enzymes BAM-H1 EcoR1 and ampicillin containing nutrient agar for selection? ORI and AMPR represent origin of replication and gene for amplification respectively. Now for any vector there should be an origin of replication. Now if you cut the origin of replication, the plasmid is not going to divide. Or if the origin of replication is lost, then also the plasmid is not going to divide and you will not get any recombinant clones. So first condition, if you see here you have EcoR1, here you have BAM-H1, you will cut it over here. This portion will be removed and in place of that your gene of interest will be incorporated. There will be ampicillin resistance for selection, ORI for replication. So this plasmid vector would be the right choice. If you see this plasmid vector, here is BAM-H1, here is EcoR1, you cut it, your ampicillin resistant gene is going to be lost. So selection will not be possible on an ampicillin containing nutrient agar. So that is not possible. Now here if you see here BAM-H1 is removed, EcoR1 is acting. Here is your ORI. Now the ORI will be lost. That is your origin of replication itself is lost. So the plasmid is not going to be divided. So that cannot be used for cloning the gene. Now here the BAM-H1 is cutting at your origin itself. So origin itself is lost. So no replication. So this also cannot be used. So the answer here is option A, that is this plasmid. Then next one. Polymerase chain reaction is used to amplify a gene of interest if after 30 cycles of PCR 1 billion copies, that is 10 raised to 9 of gene of interest are produced, approximately how many copies of gene of interest were present at the end of the 20th cycle? We will do the backward calculation. So 30 cycle it is 10 raised to 9. 29 cycles will be 10 raised to 9 divided by 2. 28 cycles will be 10 raised to 9 divided by 2 raised to 2. So it will be 4. The 27 will be 10 raised to 9 divided by 2 raised to 3. So it will be 8. And so on and so forth. We will get at 20 cycles 1 by 2 raised to 10. So 1 by 1024, that will be approximately 1 million. So the answer here is option A. Then next one. A population with n is equal to 400 individuals increases in number till it reaches an asymptote of k is equal to 500 individuals, k being the carrying capacity. Assuming the intrinsic rate of natural increase to be 0.01, what would be the population growth rate dn by dt? So dn by dt is equal to r capital N 1 minus n by k, that is the formula for dn by dt. They have given the values, n is equal to 400, k is equal to 500, small r is equal to 0.01. Substitute the values and you will get it as 0.8. Answer is option A. Then next one. Which of the following statement is correct? Hemichordata is not considered as a chordate subphylum and possesses a protochord called stomochord. So Hemichordata, it is not a chordate subphylum. It is a phylum of marine animals that is closely related to chordates but cannot be considered as subphylum Chordata. So this statement is correct. Option A. In other statement, is a subphylum under Chordata. Option B is a subphylum under Chordata. So B and C are not correct. Then Hemichordata is not considered a chordate subphylum because it possesses water vascular system. Water vascular system is found in echinoderms, not in hemichordates. So D is also not correct. So the answer here is option A. Then next one. Which of the following statement is correct about oxygen dissociation curves relative to curve B? So this is curve B. This is A and this is C. So curve A represents favorable oxygen association with hemoglobin at low H plus concentration. Low H plus concentration means under alkaline conditions. So this means hemoglobin under alkaline condition, hemoglobin has higher affinity for oxygen. Oxygen binding to hemoglobin is increased, but oxygen released to the tissues is decreased. So A is correct. Curve A represents favorable oxygen association with hemoglobin at low pH plus and the curve shift to the left. So when compared to B, this is towards the left side. So A is correct. Now if you see curve C. Curve C represents favorable oxygen association with hemoglobin. No. Here it is not favorable oxygen association because oxygen is decreased in case of C when carbon dioxide concentration is increased. So favorable with hemoglobin at low partial pressure carbon dioxide. No. B is not correct. Curve A represents favorable oxygen association with hemoglobin at low pH. Not at low pH, it should be high pH. And low H plus concentration. So C is also not correct. Curve C represents favorable oxygen association. It is not favorable association. At high PCO2, there is no high oxygen concentration at curve C. C represent high carbon dioxide level and it shifts the curve to the right. So the answer here is option A. So that's it. Thank you for watching my channel. Please do like, share and subscribe. If you are new to my channel, please don't forget to subscribe. Then if you want still previous year question papers of IAT to be answered, I have already done that in my channel playlist. My channel name is Omni Bio Entrance. You can go to the channel, see for my playlist and you will find the question papers there. Thank you for watching my channel. Please don't forget to subscribe. Thank you.